Hello, everyone. Our lesson for today is about foundations of microbiology. In the first part of this lesson, you will learn about the works of various people that led to the defeat of the spontaneous generation theory, as well as the defeat of the different theories that explain the evolution of the first true cells. In the second part, you will learn about the contributions of various scientists that led to the establishment of the field of microbiology. Thus, at the end of this lesson, you will be able to describe the origin and evolution of life and describe the history of microbiology. A motivation question for this lesson is this: what would have happened today if the field of microbiology was not established? So at the end of this lesson, I hope you will be able to answer this question. Now, first, we will talk about the origin and evolution of life. So, a long time ago, it was Aristotle who proposed the theory of spontaneous generation, otherwise known as abiogenesis. This is an idea that living things can arise from non living matter. For centuries, people based their beliefs on their interpretations of what they saw that is going on in the world around them without testing their ideas. They did not use the scientific method to arrive at answers to their questions. Thus, their conclusions were just based on untested observations. Now, this idea lasted for almost 2,000 years. Now, what are examples of spontaneous generation? So, before, their observation is that every year in the spring, the Nile River flooded areas of Egypt along the river that leaves behind nutrient-rich mud that enable the people to grow that year's crop of food. However, they noticed that along with the muddy soil, there's a large number of frogs that appeared there that weren't around during the drier times. So it was perfectly obvious to people that the muddy soil are the ones that gave rise to the frogs. Another example is based on observation that since there were no refrigerators back then, the mandatory daily trip to the butcher shop, especially in summer, meant that they will encounter flies around those meat. Typically, the carcasses of this meat were hung by their heels, and customers just selected which chunk that the butcher would carve off for them. And based on their observation, that obviously the rotting meat had been hanging in the sun all day was the source of the flies. However, not all people were convinced about this idea that non-living matter can give rise to a living matter. They believe that a living matter can only come from another living matter. So life begets life. Okay. And so the quest to disprove spontaneous generation has started with the work of Francesco Redi. In 1668, Francesco Redi, an Italian physician, did an experiment with flies and wide-mouthed jars containing meat. Redi used open and closed flasks which contained the meat. So here is a closed flask and this is an open flask. His hypothesis was that the rotten meat into flies. He found that if a flask was closed and the lid it is tight around the mouth of the jar, adult flies could not get in And so no maggots would develop on the rotting meat inside. Now, in a flask without a lid, maggots soon were seen in the meat because adult flies had laid eggs, and more adult flies soon appeared. So, because of this experiment, Redi was able to demonstrate that it is not the flies the meat that gave rise to the flies, but the flies that lay their eggs on the meat, and the eggs hatched and became the flies. The results of this experiment disproved the idea of spontaneous generation for larger organisms, but now people still thought that microscopic organisms like algae or bacteria could arise spontaneously. And so the quest for disproving spontaneous generation for microorganisms has started. Now, one of the pioneer is Anton van Leeuwenhoek, or other books would tell you Antony van Leeuwenhoek or Antoine van Leeuwenhoek. So Leeuwenhoek began making and looking through simple microscopes. Now, these microscopes he made on his own. He examined water and visualized tiny animals, fungi, algae, and single-celled protozoans, which he called animal kills. And by the end of the 19th century, these organisms were called microbes. This is the a picture of the first microscope that Leeuwenhoek had created. These are some of the illustrations that were made by Leeuwenhoek. Another scientist was John Needham. He showed that microorganisms flourished or appeared in various soups that have been exposed to the air. So he claimed that there was a life force that is present in the molecules of all inorganic matter, including air and the oxygen in it, that could cause spontaneous generation to occur. So Needham's experiment seemed to support the idea of spontaneous generation. That was his idea, that there is a life force in the air, and you expose something on it, then from these soups, microorganisms would appear. So what he did, he first heated the broth, and then to kill all the organisms, and then he exposed it to air, and voila, now there was microbial growth. But what John Needham did not realize was that bacteria were already present in Needham's soup, but he already boiled it. He did not boil it long enough to kill all the microorganisms. Not so. It may seem to support spontaneous generation, but actually it is not. So Lazzaro Spallanzani extended the work of Needham. What he did was he boiled the soups for almost an hour and sealed the containers by melting the slender necks of these glass containers closed. So for a long time, the soups remained clear. Later, he broke the seals, and the soups became cloudy with microbes. So, proving that the microorganisms was already present in the Needham soup, and he did not just boil it long enough. However, critics said that sealed vials did not allow enough air for organisms to survive, and that prolonged heat had destroyed the life force, of course. So, the air is needed in order for the microorganisms to spontaneously grow. Okay, in the soup. That was what people still believe. Therefore, spontaneous generation for the microorganisms still remained the theory of the time. And so, the theories finally changed when by 1860, There was a great debate about this, and the Paris Academy of Sciences offered a prize for any experiments that would help resolve the conflict. And the prize was claimed in 1864 by Louis Pasteur. He published the results of an experiment that he did to disprove spontaneous generation in microscopic organisms. So he hypothesized, no, that the microbes come from the cells of organisms on dust particles in the air and not the air itself, because that's what people believe. No, that people believe that Lazzaro Spallanzani's experiment did not prove anything because he sealed the flask, and when he opened it, because of the air, then the microorganisms spontaneously has grown. So what Louis Pasteur proved is that the microorganisms is not from the air itself, but from the dust that is carried on, no, by the air. So Pasteur, he put both the broth into several flasks, which he called the S-shaped flask or the swan-necked flasks. Each flask was boiled, no, and not sealed because that's what people wanted. Air should come in, okay, and placed at various locations. So this is example of the S-shaped flask that Louis Pasteur has created. So it would allow the air to still come in. Okay. So what he did was the soup or the culture media was heated just like what Lazzaro Spallanzani has did. He boiled it long enough and he let the flask sit for a while. And for a long time, no bacteria or no microbial growth was observed, even though air could still come in. Okay? And when he removed the neck and let it sit, then bacteria flourished into the soup. Or when he tilted the flask so that the soup or the culture media would come in contact with the mouth of the flask, microbial growth was present. So this is the experiment that proved that microorganisms are not born from the air, but microorganisms were already present in the dust particles in the air, and that the soup became a conducive environment for the growth of this microorganism. Okay. Now, what's remarkable about this experiment is the shape of the flask. Because of the shape of the flask, it allowed the air to come in, and the dust from the air to be deposited at this part, because the dust cannot, because the dust is heavy, and so the air can only the air can rise and go inside the flask, not the dust, because they are very heavy. They could be deposited at this part of the flask. So this experiment again proved that microbes only come from other microbes, or life from life. And this is what we call biogenesis. Now, if life only comes from another life, the question is, where did the very first life came from, or that very first true cells? Now, where did it came from? Now, the theory of Panspermia tells us that the seeds of life exist all over the universe, and that it could be propagated through space from one location to another. So this theory is telling us that life on Earth may come from the universe, not from space. Some believe that life on Earth may have originated through the seeds, and that they are brought in on asteroids and comets that have bombarded the Earth. Now they said now that a very good example of an organism that may have come from space now is the bacteria called Deinococcus noredeodorans. Now this is a highly radiation resistant bacteria. No extreme this bacteria could tolerate or could resist extreme radiation that is only present no in the space. So this could possibly be no the link no to that or an evidence of that theory. Another evidence could be the presence of carbonaceous chondrites. Thus, it represents some of the most pristine matter known, and their chemical compositions match the chemistry of the sun more closely than any other class of chondrites. So this proves that there are bombardments that happened no, from space no, into the Earth. Another theory no, that would describe the origin of life is called abiotic chemical evolution. Okay, so people or some scientists believe that the first cells no has arise spontaneously. Okay, from those inorganic elements, inorganic compounds present in the primitive Earth, due to energy from lightning, from UV radiation, from intense heat, there was synthesis and accumulation of organic compounds. From those organic compounds, uh, larger molecules polymerized and aggregated and gave rise to heredity. Okay, from there, the first true cells were formed, and these cells were capable of cellular respiration and photosynthesis until the types of or the present types of cells emerged. Now, the very first aggregate of these polymers or compounds, organic compounds, that is believed to be the precursors, the one that evolved first before the first two cells. No, they're called the precursors to the first two cells. Are called the protocells. They're also called protobiont. This is a self-organized and endogenously ordered spherical collection of lipids. They propose as a stepping stone to the origin of life. And so they believe because of the formation of these, they look like bubbles. The polymers that are aggregated inside, gave it the environment inside these protocells gave it a conducive environment for the heredity to flourish. Okay. Now, I won't talk much about the details of this evolution, but from these precursors to the first two cells rose, or gave rise to the last universal common ancestor. So this universal common ancestor, they say, or what we call, is believed to be the origin of all life on Earth. These are otherwise known as the progenotes. The last universal common ancestor, the LUCA, otherwise known as the progenotes. Now, sometime in the history of the Earth, these progenotes became separated. In space, one went to other parts of the Earth, other went to other parts of the Earth, and we know that the Earth has different environmental conditions. And so these environmental conditions may have provided an environment for the different types of evolutions that these progenotes would undergo. In other parts, these progenotes became pre-Archean. The other parts, these progenotes became the pre-bacterium and evolved later on to become the cells of the bacterial lineages. Others, the pre-Archean, became the cells of the Archean lineages. But sometime in the history, a pre-Archean, a pre-bacterium, has fused. So they fused, they came in contact, and they produced no pre-eukaryotic cells. These later on, the pre-eukaryotic cells now gave rise to the present types of cells that we have. So when we go to during our when you go to cell and molecular biology course, now we will explain how these pre-eukaryotic cells would become later on the cells of the plants, the animals, the protists, and the fungi. So for now, we just know that a fusion of a pre-Archean and a pre-bacterium produced a pre-eukaryotic cell. Now, some of those cells that were not involved in the fusion, again, became the true archaeal cells, and those pre-bacteria became true bacterial cells. These two, the archaea and bacterial cells, are prokaryotic cells, meaning they do not have their own distinct nucleus, while the pre-eukaryotic cells evolved and became or developed its own distinct nucleus. Okay, so that's how, no, that's what people, that's the most acceptable, no, theory, no, that scientists, no, believe as of the present, no, for the origin of life on Earth. Now let us talk about the history of microbiology. So it was Robert Hooke. No? He is an English mathematician and natural historian, and also an excellent microscopist, who published the book Micrographia, the first book devoted to microscopic observations. He illustrated, among many other things, okay, the fruiting structures of the molds, okay, or the filamentous fungi that we know now. So this was the first known description of microorganism, and so we could say that Robert Hooke is considered as the father of microbiology. You already know Anton van Leeuwenhoek. So in 1684, van Leeuwenhoek was well aware of the work of Hooke. He used extremely simple microscope on his own to construct and examine the microbial content of natural substances. He was the one who first to see bacteria in 1676. Ferdinand Cohn is trained as a botanist and became an excellent microscopist. He studied unicellular algae and bacteria, particularly, and he was not particularly interested in heat resistance in bacteria. So that made him discover that some bacteria will form in the spores. So later on, when we go to module 2, we'll talk about endospores. So basically, endospores are these cell structures that are produced by some type of bacteria when they are exposed in a condition that is not conducive for their growth. And so what happened is that if they send something in the environment that it may be harmful to them, so they will enclose their genetic material in a very hard covering and will produce it, and then these endospores will become dormant for many years. Ferdinand Cohn also was the one who described the life cycle of endospore forming bacterium, the Bacillus. And he showed that vegetative cells, but not endospores, were killed by boiling. He was instrumental in laying the groundwork for a system of bacterial classification, including an early attempt to define a bacterial species. Of course, Pasteur and the defeat No, and how he defeated spontaneous generation. No, is also very he is very his works were very relevant for the establishment of the field of microbiology. And what are the important discoveries that we gained from Pasteur's experiments? So now we know that microbial life can be destroyed by heat. So with that, aseptic techniques were developed. So aseptic techniques, these are techniques that prevent contamination by unwanted microorganisms. So in the laboratory, you will be learning how to do aseptic procedures in the laboratory. And also the concept of pasteurization. So pasteurization is commonly employed in milk. So when they subject milk to a certain temperature, just enough to eliminate majority of the microorganisms that causes spoilage, but not too much that will degrade the property of the milk. That is pasteurization. It is different from sterilization, because in sterilization you can use heat to eliminate all or to kill all of the microorganisms that is present in a particular sample, in a particular substance, uh, in a particular equipment. Okay. Now, another important person in the history of microbiology is Robert Koch. So, in his early work, Koch studied anthrax, a disease of the cattle and occasionally of humans. So, anthrax is caused by an indus-forming bacterium called Bacillus anthracis. And by careful microscopy and by using special stains, Koch established that the bacteria were always present in the blood of an animal that was succumbing to the disease. However, he reasoned that the mere association of the bacterium with the disease was not proof of cause and effect. No? If you compare it to a particular crime, for example, just because a person is found beside a dead person does mean that that person was already the one who killed the other person. So the mere association of a bacterium with a disease was not proof of cause and effect. That's why he studied cause and effect experimentally using anthrax, and he developed these following postulates. So this is what we also call pathogenicity testing. Okay. So how will you prove the pathogenicity of a particular microorganism? When can you say that it is that particular microorganism that is causing the disease? Okay, so first, the microorganism must be found in abundance in all organisms that is suffering from the disease, but should not be found in healthy organism. So if you try to say for example, investigate a new occurrence of nangamatay ang mga baboy, nangamatay ang mga cattle, okay? And you don't know what is the causative agent, so you must as well take samples from the disease, the one that namatay, and from the healthy, so that you compare which is present in the disease that is not present in the healthy animal. So those that are unique to the diseased organism must be isolated. So that is now what we call the suspected pathogen. Suspected pa lang. No? So the microorganism must be isolated from a diseased organism and must be grown in pure culture. No? So you just hope that the microorganism that you are dealing with is culturable. Otherwise, you will never determine it. Okay? So you have to isolate it in pure culture, and then from the pure culture, the microorganism should cause disease when it is introduced into a healthy organism. Okay. Now, when the healthy organism has also died, also showed the same symptoms from the original, okay, it must be re-isolated and must be found to be the same as the original. Okay, so this is how you do the normal or the classical pathogenicity testing based on Koch postulates. So again, when we say pathogen, we are referring to microorganisms that are able to cause disease or the disease-causing microorganism. But when you say virulence, you are referring to the degree to which a particular pathogen causes disease. So do not confuse yourself with the term pathogen and virulence. So if you want to understand or to want to prove the pathogenicity of a particular microorganism, you have to do this. Now, there are just some drawbacks or some disadvantages of using this because not all microorganisms, just like what I said earlier, are culturable. Okay? And also, not all microorganisms, when they are cultured in vitro, in the laboratory, and when they are infected into the other animal, others would lose, other microorganisms would lose their virulence factors when they are already in culture. So, maski siya pa to ang tinood nga pathogen when they are infected into a particular healthy animal, basig dili siya mag-cause of disease because he may have lost or it may have lost its pathogenicity during culture. But that is very rare also. So there are some drawbacks when you are using this. So your target, you must understand your target microorganism well. Okay. Robert Koch was the first person to culture bacteria on a solid culture media. No? So he has a lot of contributions in microbiology. Initially, he used potato slices. Okay? However, not all bacteria can grow on it, and slices are frequently overgrown with molds. So it is not really a good culture media. So he also used gelatin, the gulaman, as a solidifying agent for various nutrient fluids. Although this nutrient supplemented gelatin was a suitable culture medium for the isolation and study of various bacteria, the thing is that it did not remain solid at 37 degrees Celsius. And that's not good because if you are studying human pathogens or animal pathogenic microorganism, you must be able to grow them in a solid media at 37 degrees Celsius because that is the optimum temperature to where they grow because that is the normal body temperature. Okay? So, it is not really a good solidifying agent. So with this, development such as the production of agar escalated. So agar is a polysaccharide derived from red algae. It's a desirable gelling or a solidifying agent for microbial culture media up to this day. Now let us emphasize that the agar itself is not the culture media. Okay? The culture media, the media are those nutrients that you put in to your media itself. The agar is just a solidifying agent, meaning you just added it in order for the media to solidify because it is important or so solid culture media are very important in many research. Okay, but the culture media is not the agar itself. It is just a solidifying agent for culture media. Okay, so Walter Heath is an associate of Koch, was the first to use agar in bacterial culture. Okay. Do not confuse yourself with the contribution of Robert Koch. Robert Koch was the first person to culture bacteria on a solid culture medium using potato slices. But it was Walter Heath who first used agar as in bacterial culture. That was a suggestion from his wife Fanny, who used agar to solidify fruit jelly. Agar is an excellent solidifying agent because, unlike gelatin, it remains solid at 37 degrees Celsius, and after melting during sterilization process, it would remain liquid at about 45 degrees Celsius. That's important because you need to handle the the molten agar at a liquid phase to manipulate it in the laboratory. No? So at 45 degrees Celsius, it will be easier to handle, no, the agar. Also, agar is not degraded by most bacteria and typically yields a transparent medium, which is important in bacterial classification to differentiate one colony from another and for characterization of different colony shape, structure, and color. Okay. In 1887, another scientist, Richard Petri, a German bacteriologist, he developed transparent double-sided dishes that bear his name as modification to Koch's flat plate technique. So he called it the Petri dishes. So the advantage of Petri dishes were immediately apparent. So because Petri dishes could be easily stacked on one on top of the other and sterilized separately from the medium, and following the addition of molten culture medium to the smaller of the two dishes, the larger dish could be used as a cover to prevent contamination. At the same time, there would be a space no for air to go inside no the media without direct exposure to contaminants. Now, why is pure culture significant? Why is it important? So it was culture realized that when you culture the microorganism, you will find that the differences. So you have here a microorganism that is cultured in different culture media, and he said that different microorganisms show these different characteristics. No, just it could be it could be the same, just like in plants and animals. In plants, in classifying plants, you can classify them based on their flower, based on the leaf arrangement, based on color, root, no? So just like that, microorganisms, you could also use these characteristics to classify microorganism. Now that's why it's very important to culture the microorganism. So the rise of microbial diversity. So it was Martinus Beijering who formulated enrichment culture technique. An enrichment culture technique is when you use a specific condition to highly favor a specific microorganism from a natural sample. So, for example, I want to isolate nitrogen fixers from the environment, from the soil. So, in order to favor the isolation of nitrogen fixers, I will put something in my culture media. So that is now called an enriched culture media. So enrichment culture technique. No? Isolation of microorganisms from natural samples using highly selective techniques of adjusting nutrient and incubation conditions to favor a particular metabolic group of organism. So Martinus Beijering was the one who isolated nitrogen-fixing bacterium Azotobacter from soil. He also isolated many soil and aquatic microorganisms. He also studied tobacco mosaic diseases. No? So he used selective filtering techniques to show that the infectious agent was smaller than the bacterium and that it somehow became incorporated into the cells of a living host plant. So that insightful work was done by Beijering. However, he was not able to realize it that those were already viruses. But the basic principles of virology was already established, was believed to have come from his idea. Sergei Vinogradsky. So he was the one who proposed chemolithotrophy. So chemolithotropy is the oxidation of inorganic compounds to yield energy. Unlike humans, so we know that we only consume organic compounds in order for us to produce the ATP for our energy. But microorganisms that not just use organic compounds, there are also groups of microorganisms who can use inorganic compounds in order for them to gain energy. He further showed these organisms, which he called chemolithotropes, obtain their carbon from carbon dioxide. Vinogradsky revealed that, like phototrophic organisms, there are also chemolithotropic bacteria that were autotrophs. So they're actually called chemoautotropes, not photoautotropes. because phototrophic organisms, they use uh, sun or energy or light energy as energy source. But for chemolithotropic bacteria, the oxidation of inorganic compounds yields energy that will make them or you make them able not to produce organic compounds. And he was the one who first to isolate a nitrogen fixing bacterium, not the anaerobic Clostridium pasteuriano. Okay? So that ends our lesson no, on the origin and history of microbiology, origin of life and history of microbiology. So when you review no, our lesson, you should be guided no, by this different question. What is spontaneous generation theory and how was it defeated? So you take note of the different people that are involved in the defeat of spontaneous organisms for larger organisms as well as for microorganisms. And who are the people behind the establishment of the field of microbiology? So you need to remember them and what are their specific contributions. Okay. Okay, so that ends our lesson on the foundations of microbiology. Thank you very much for listening. If you have any question, you can contact me through my email. You can also message me through our group chat on Facebook Messenger. Okay, thank you again and see you next time.